Hello everyone, I'm Dr. Sina Shamekhi, and this is the third lecture of the Bioinstrumentation course. We will talk about biopotential electrodes in this lecture. First of all, let me consider the introduction. In order to measure and record potentials, as some ionic currents, in the body, it's necessary to provide some interface between the body and the electronic measuring apparatus. Current flows in the measuring circuit for at least a fraction of a period of time over which the measurement is made. And also it's important to know bipotential electrodes is a transducer that converts the body ionic current, remember that, in the body into the traditional electronic current flowing in the electrode. It means that the ionic current generated inside our tissues, our excitable cells, will be converted to electronic current using our electrodes, our bipotential electrodes. Current is carried in the body by ions, whereas it is carried in the electrode and its lead wire by electrons. Na, K, and also Cl ions is the main carriers for the body ionic current, and inside our wires electron will carry the current. Let me describe some basic chemical information, some basic chemical requirements about the electrode before talking about the details of a practical fabricated electrode. We have two main reactions, oxidation and reduction. Oxidation reaction causes atom to lose electron. Oxidation causes atom to lose electron. And reduction reaction causes atom to gain electron. As you can see, oxidation, oxidant plus an electron in reduction reaction generates the product. In the product, oxidation number will decrease and the number of electrons in the product will increase. And in oxidation reaction, the reductant will lose electron. Lose electron and oxidation number will increase. As you can see at the bottom of this slide, we have two main figures. We plotted for description, the oxidation and reduction reaction. And as you can see, we have a metal inside an electrolyte, a metal inside a volume of an electrolyte. And also here, metal and electrolyte. First of all, assume that the current flow is from the left to the right, from metal to electrolyte. The current flow and the flow direction of electron are opposite. It means that if the current flow is from the left to right, the direction of the electron is from the right to left. Therefore, the metal atom should lose its electron to give the electron to the wire connected to this metal. We have a connection of metal to this wire to this metal. And when we are talking about the current flow from the metal to electrolyte, it means that the electron should be travel at the opposite direction. Okay, after losing the electron, we have M plus ion, and M plus ion as a cation can react with anion. M and also M plus A minus will generate Ma deposit. As you know, we have some problem in pen. And as you can see, also M loses his electron, and anion also share his electron with M plus. It means that A also loses its electron. And losing electron is an oxidation reaction. Therefore, oxidation is dominant when current flow from electrode to electrolyte. And if the current flow is from the electrolyte to the metal to the electrode, it means that the direction of the electron is at the direction of the electron is from the metal to electrolyte. Therefore, we need to gain electron. Cations should gain electron come back home, and also the deposit Ma will be divided to M plus A negative. And as you can see, M gains its electron and also A gains its electron too. Therefore, as you can see, when an oxidant gains its electron, reduction reaction is dominant. Therefore, reduction dominate when the current flow is from the electrolyte to the metal. When an electrode is placed inside an electrolyte, because of the concentration of ions inside the metal and inside the electrolyte, we have a specific potential. We called it half cell potential. And this half cell potential is determined by metal involved, the type of the metal, concentration of its ion in solution, temperature of the electrolyte, and other second order factor. And this half cell potential is based on a certain mechanism. This certain mechanism separates charge at the metal electrolyte interface, results in one type of charge is dominant on the surface of the metal, and the opposite charge is concentrated at the immediately adjacent electrolyte. As you can see in this slide, each metal has its own half cell potential value, and all of these values are measured based on a standard algorithm, a standard method based on the measuring the potential between the half cell of these metals and another half cell consists of hydrogen electrode. And we assume that the hydrogen electrode has a zero voltage, zero half cell potential voltage, and all other half cell potentials are measured, refer to this standard hydrogen electrode, and now we have standard values of half cell potentials. And also, you remember this box. the more negative slash positive the half cell EMF, the half cell potential, the greater slash smaller the tendency of the reductant to donate electrons. Donate electrons means oxidation. And the smaller slash greater the tendency of the oxidant to accept electrons. The more negative slash positive half cell potential, the smaller slash greater the tendency of the oxidant to accept electrons. Accept electrons means reduction.